Imagine waking up tomorrow morning, right, and you find out that the microscopic bugs living inside your walls have just completely rewritten the architectural blueprints of your house.
Yeah, like they just picked up the kitchen and moved it.
Exactly. They moved the kitchen, they rezoned the bedrooms, they completely altered the structural integrity of the whole building.
Which sounds completely absurd.
It sounds like a bizarre science fiction scenario, but according to a really groundbreaking 2026 study published in the journal Cell, that is almost exactly what is happening inside your body right now.
It is. And, it's fascinating because it completely dismantles a core assumption of human biology.
Right, which is why we're doing this deep dive today. Our mission is to fundamentally change how you think about your gut. So we're looking at this paper by Rispol and colleagues.
Right. It's titled Microbiome-Produced Nicotinic Acid, Controlled Colon, Regional Identity, and Injury Susceptibility.
Which is a mouthful.
It is a mouthful, yeah. But the core idea is that we used to view the colon as this very binary, generic biological chute.
Like a water pipe.
Exactly.
Yeah.
The stomach and the small intestine, they do all the complex chemical absorbing, right?
Yeah.
And the colon is just this homogeneous pipe at the end of a line, just drying things out.
A simple, uniform tube. And, it's comforting to think of it that way. It's simple. But this deep dive is going to show you that the colon is actually a highly specialized factory.
Right, with distinct localized zones.
Yeah, distinct neighborhoods, proximal, mid, distal. And the truly wild part, the thing that just blew my mind, is that our own human cells don't build these zones themselves.
No, they don't.
The microscopic tenants living inside you, like your gut bacteria, they are acting as the regional managers. They are physically programming your human DNA to build specific biological neighborhoods.
Yeah, because we've known for decades that the small intestine regionalizes itself.
Right.
It has this intrinsic hardwired genetic programming that just tells its cells what to become.
It's self-sufficient.
Exactly.
Yeah.
But this paper reveals that the colon outsourced that job. It relies almost exclusively on the microbiome to tell its tissue what to do, what to be, and how to protect itself.
Which just fundamentally changes how we think about digestion and antibiotics and even chronic illnesses like Crohn's disease.
Definitely.
So to prove that bacteria are secretly managing our cellular architecture, the scientists had to do the biological equivalent of, firing the managers and seeing if the factory falls apart.
Right. They had to map the colon with and without bacteria.
Which brings us to our first segment, the myth of the homogeneous colon and wiping the map. So how do they actually map this out?
Well, the way they achieve this mapping is a marvel of modern biology. It's called single-cell RNA sequencing, or cRNA-seq.
Okay, break that down for us. What is that?
So they didn't just take a biopsy, blend it into a smoothie, and look at the average genetic material.
Right, which is what we used to do.
Yeah, exactly. Instead, they zoomed in on the entire colonic and cecal epithelium of mice, and they cracked open millions of individual cells, one by one.
Wow, millions of them.
Yeah, and they're essentially reading the live order slips inside each cell. They look at the messenger RNA, the mRNA, to see exactly which proteins that specific cell is currently manufacturing.
So it's like going into a city and reading the grocery receipts of every single resident to figure out what kind of neighborhood they live in.
That is a great analogy.
Yeah.
And by doing that, they established a baseline in normal, healthy mice.
Okay.
And they found that there isn't just one generic type of colon cell. The map showed distinct genetic signatures.
So clear zones.
Very clear.
Yeah.
Defining a proximal colon, a mid-colon, and a distal colon. Distinct zones with distinct jobs.
Okay, so then came the real test. The researchers had to see what happens when you wipe out the bacteria, and they looked at two distinct models for this, right?
Right, 2 models where the bacteria were removed. First, they used germ-free mice.
GF mice.
Yes. These are mice born via C-section and raised in completely sterile, sealed isolator bubbles. So they have 0 microbes. Absolutely none.
Never even exposed to a single bacterium.
Exactly. And the second model was normal mice that were given heavy doses of broad-spectrum antibiotics.
Oh, so they wiped out the existing established gut bacteria. How long did they give them the antibiotics?
For either 4 or 14 days,
Hmm.
just to really clear the system.
Okay, so what happened to the map when the microbes were gone?
In both of those bacteria-free models, the specialized zones just vanished. The genetic expressions completely blurred together. Without the bacteria there to provide localized instructions, the colon lost its regional identity.
So the proximal, mid, and distal cells, they just reverted into one confused, uniform blob of generic tissue.
Exactly. And the researchers tested this across entirely different genetic strains of mice, too, like C57BL6 and CD1.
Why does the strain matter?
To prove this isn't a quirk of one specific lab breed, it proves it is a universal mammalian rule.
Wait, I have to challenge the logic of this. I mean, from an evolutionary standpoint, that seems like a terrible design flaw.
How so?
Well, if having these specialized zones is so critical for a functioning gut, why didn't human and mammalian stem cells just hardwire the blueprints into our own DNA?
Like the small intestine did.
Yeah. Why outsource the management of our own tissue to unreliable bacteria? I mean, if I get food poisoning or take an antibiotic, my whole system crashes.
It does sound incredibly risky. But it's actually an evolutionary masterclass in adaptability.
Okay, explain that.
So the small intestine deals with a relatively consistent chemical environment, right? It's mostly stomach acid and digestive enzymes.
Pretty standard stuff day to day.
Right. But the colon is the fermentation chamber. The materials arriving there change drastically depending on what you eat.
Oh, I see.
Yeah. Whether you ate a heavy fiber diet, a high protein diet, or say you survived a famine, the environment shifts wildly.
Right.
By outsourcing the architectural management to the microbiome, the colon remains dynamic. The bacteria sense the changing nutritional environment and adjust the cellular zoning accordingly.
Ah, so it's flexibility over rigidity. It's an adaptable software update instead of hard-coded hardware.
Exactly. It's a brilliant survival strategy.
That makes so much sense. Okay, so let's do a quick recap before we move on. We just hit the 5-minute mark, so let's synthesize this.
I'm good.
Bullet point 1. Your colon is not a generic tube. It has specific regions like proximal, mid, and distal. Bullet point 2. Your microbes actually control the creation of those regions. And bullet point 3, wiping out your microbes with antibiotics literally deletes those specialized zones and blurs your colon into a generic blob.
That sums it up perfectly.
So with that in mind, here's a question for you listening. Think about times you've taken broad-spectrum antibiotics. How might temporarily wiping your internal cellular GPS impact your overall digestion? It really makes you think about your medical history differently.
Oh, it really does. The GPS goes completely offline. The cells physically lose their bearings.
Which leads us perfectly into our second segment, the secret messenger. So if the GPS is offline without bacteria, how do the microbes actually transmit the coordinates when things are healthy?
Right. Because bacteria don't have a nervous system.
Exactly. They can't plug into our brains or send an email. They communicate purely through chemistry.
And to intercept that chemical communication, the researchers utilized a technique called untargeted metabolomics.
Untargeted metabolomics, meaning they didn't know what they were looking for.
Exactly. Instead of looking for a specific known molecule, they cast a massive net to catalog the entire chemical soup inside the gut.
How many chemicals are we talking about?
They analyzed 2,288 different metabolites across the varying regions of the colon.
A lineup of over 2,000 molecules. They were basically auditing every single byproduct the bacteria were churning out.
Yeah, and one chemical anomaly stood out drastically. They discovered that a specific molecule called nicotinic acid, which is abbreviated as Na, was highly concentrated in the proximal colon.
Okay, so Na is in the proximal colon. But what happened to it in the antibiotic mice?
In the mice treated with antibiotics, the local levels of nicotinic acid plummeted to almost nothing.
Wow.
But here is the biological smoking gun they found. While the nicotinic acid vanished, its precursor molecule, which is nicotinamide, actually spiked in the tissue of those same antibiotic-treated mice.
No. Okay, so it perfectly illustrates an interrupted assembly line.
Yes, exactly.
The host body provides the raw material, the nicotinamide, and the microbial workers take that material and convert it into the finished product, the nicotinic acid.
You've got it.
So when you kill the workers with antibiotics, the raw material just piles up on the conveyor belt and the final messenger is never produced.
That is exactly what the metabolomics data proved.
So the bacteria are manufacturing nicotinic acid, and that Na serves as the physical zoning permit for the proximal colon. But how does a chemical floating around in the gut actually force a human cell to change its physical identity?
This is where we get into the microscopic mechanics, the actual lock and key of the system. Okay, lay it on me.
The nicotinic acid permeates the human colon cell and travels to the nucleus. And there, it finds a very specific receptor called PPAR alpha.
PPAR alpha.
Yes. That stands for peroxisome proliferator activated receptor alpha.
Okay, so think of PPAR alpha as a dormant physical keyhole built right into the human DNA.
Right.
It sits there doing absolutely nothing until the bacteria forge the exact chemical key, the nicotinic acid, to fit into it.
Exactly. And when the NA key turns the PPAR alpha lock, it activates a cascade of specific proximal genes, things like HMGCS2 and CBR3.
And the researchers actually proved this, right?
They were meticulous in proving this mechanism. They grew mouse organoids.
Which are like mini guts.
Yeah, they are living 3D miniature structures of intestinal tissue grown in a petri dish. And they applied nicotinic acid directly to these mini guts, completely free of any live bacteria.
Oh, so just the chemical.
Just the chemical. They also tested a synthetic drug called WY14643, which is artificially designed to turn that exact PPAR alpha keyhole.
And what happened? In both scenarios, the human cells instantly started expressing the proximal genes.
Wow.
The chemical alone was enough to make the cells adopt their specialized identity.
But they took it even one step further, right? With the knockout mice.
Yes, the conditional knockout mice, the PPARA CKO model. This is brilliant. They genetically engineered mice with a targeted weakness. By giving the mice a drug called tamoxifen, the researchers could trigger an active deletion of the PPA alpha receptor in the live colon tissue.
So they essentially erased the keyhole while the mouse was alive.
Exactly. And when they did that, the colon completely lost its proximal identity.
Even though the bacteria were still there.
Even though the bacteria were still there, furiously churning out nicotinic acid. The key was present, but the lock was gone.
Which brilliantly proves the symbiosis. You absolutely need both the bacterial key and the host's lock to build a neighborhood.
You do.
But, this brings up a highly practical question. Nicotinic acid isn't some exotic, undiscovered chemical. It's literally a form of vitamin B3.
Yes, it is.
I could walk into a pharmacy right now and buy a massive bottle of it. So if someone's colon is struggling to maintain its healthy zones, could they just swallow a handful of B3 to manually turn the key and fix their colon's GPS?
It is the most logical next thought. And the scientists absolutely anticipated it.
Did they test it?
They did. They performed an oral gavage experiment on a mice, delivering a massive dose of nicotinic acid, about 600 milligrams per kilogram of body weight.
That's a huge dose.
Huge. And the NA successfully survived the stomach acid and made it to the colon.
But I'm guessing it didn't magically restore the perfect architectural map?
It failed completely. But the reason why it failed is fascinating. Taking the nicotinic acid orally flooded the entire colon systemically.
Oh.
They washed over the distal colon down at the bottom, just as heavily as the proximal colon up top. It completely ruined the localized specific enrichment.
Oh, I see. The distal cells down below started receiving proximal zoning permits,
Right.
which confused the genetic mapping of the entire lower gut.
Geography matters just as much as chemistry here.
Yeah.
You can't just carpet bomb the digestive tract with a supplement.
Right. You need the local bacterial managers stationed on the ground, handing out the specific chemical keys exactly where they are needed, molecule by molecule.
It highlights an incredible degree of spatial precision. When we eat, we're not just extracting calories for ourselves, we are supplying the highly specific raw materials to a localized workforce that maintains our internal architecture.
Which is mind-blowing. Okay, time for another quick recap.
Go for it.
Bullet point 1. The microbiome produces nicotinic acid, or vitamin B3. Bullet point 2. This Na is a chemical key that unlocks the PPAR alpha receptor in our cells. Bullet point 3. You can't just eat B3 to fix this because it floods the system and ruins the specific local zoning.
Exactly.
So here's a question for you to ponder at home. Knowing that a single microbial byproduct completely rewrites your cellular identity, does it change how you view the food you eat to feed those microbes? It's a heavy thought.
It really is.
So we've traced the entire mechanism, right? The microbiome processes raw materials into Na. The Na unlocks PPAR alpha. That receptor turns on specific genes to build the proximal colon. But we still need to answer the biggest question.
The why.
Yes, the why. Why did evolution design our colon to need this bacterial zoning permit? What is the actual function of this proximal neighborhood?
It is an active localized shield against disease. And we see the devastating consequences of losing that shield in inflammatory bowel diseases, or IBD.
IBD affects millions of people globally, and it is incredibly difficult to treat because it manifests so differently in different people.
Right. And clinically, IBD is heavily regionalized.
Explain that.
Well, ulcerative colitis, or UC, typically presents as a continuous inflammation that starts at the rectum and affects the distal colon.
Okay, so lower down.
Right. But Crohn's disease, CD, is notoriously patchy and heavily targets the proximal colon and the end of the small intestine.
Ah, so right where this nicotinic acid system is supposed to be working.
Exactly. And when the researchers analyzed biopsies taken from real human patients suffering from active Crohn's disease, the data showed something alarming.
What did they find?
The cells in their proximal colon had physically lost their proximal identity.
You're kidding.
No, the major genes turned on by the PPARα alpha keyhole were just shut down.
Which genes specifically? What are they supposed to be doing?
One of the most critical genes in that pathway is called HMGCS2.
HMGCS2.
Yes. When the PPARα alpha keyhole is activated, it tells the cell to upregulate HMGCS2. And this is an enzyme responsible for creating ketone bodies.
Ketones?
Yes, specifically beta-hydroxybutyrate.
Wait, ketones usually come up in the context of fasting diets, right? What are they doing in the colon?
They're providing massive, highly efficient localized energy. Think about it. The colon lining takes a severe beating from digestive toxins and waste.
Yeah, it's a harsh environment.
Very harsh. So to survive, the tissue has to rapidly regenerate its cells and constantly secrete a thick layer of mucin.
Mucin, so like a physical slime barrier.
Yes, exactly. A slime barrier that prevents bacteria and toxins from touching the vulnerable human tissue. But that high-speed regeneration and mucus production requires a massive amount of energy.
So the nicotinic acid triggers the ketones, and the ketones fuel the mucosal barrier.
You got it. That is the physical reality of the shield.
And in Crohn's patients, that HMGCS2 gene goes silent.
Right.
So the energy drops, the mucus barrier thins out, and the colon lining becomes completely exposed to inflammation and injury.
The armor is completely stripped away.
Man, that is intense. But it brings up the million-dollar question for treating Crohn's. Does the disease fundamentally break the human cells, like permanently destroying their ability to produce that armor? Or is the communication line from the bacteria just severed?
The experiment they ran to answer this is, in my view, the most hopeful breakthrough of the entire study.
Oh, tell me about it.
They acquired human organoids grown from the intestinal stem cells of 3 different human donors.
Okay.
1 healthy individual, 1 patient with ulcerative colitis, and 1 patient with severe Crohn's disease.
So they took living tissue that carries the exact genetic pathology of the disease.
Exactly. Real human disease models. They then treated all 3 of these human organoid lines with the nicotinic acid and that synthetic activator we talked about, WY14643.
And what happened? Did the disease cells respond?
They responded perfectly.
Wow.
The Crohn's tissue fired right back up. It expressed the HMGCS2 gene and began synthesizing the machinery for the mucosal shield just as robustly as the tissue from the completely healthy donor.
That is incredible. So it's like finding out the factory isn't burned down, right? The workers are just sitting around in the dark waiting for a shipment of NA that never arrived.
That is a perfect way to put it. It definitively proves that the inflammatory disease does not irreparably break the internal machinery of the human cells. The cellular hardware still knows exactly how to be healthy.
The disease is just missing the bacterial signal.
Exactly.
Which is such a massive paradigm shift. Right now, our primary strategy for treating severe IBD is prescribing heavy immunosuppressants.
Right. We essentially turn off the body's entire immune system to stop the inflammation.
Which leaves patients vulnerable to a whole host of other infections. But if we can just deliver the precise signal to them, they will go right back to building the shield themselves.
Yeah, this research opens the door to a completely new frontier of personalized medicine. Instead of fighting the fire by shutting off the immune system after the mucosal armor is already broken.
We could theoretically prescribe targeted, locally delivered microbial signals.
Exactly. We can instruct the patient's own cells to rebuild their natural armor.
The therapeutic potential is just massive. We are looking at a future where we treat the gut not as a defective organ, but as a mismanaged ecosystem. We just need to reintroduce the right managers with the right zoning permits.
That's the dream.
Okay, let's do our final quick recap of this segment before we wrap up.
Sounds good.
Bullet point 1. The proximal identity acts as an energy-intensive shield powered by ketones. Bullet point 2. Crohn's patients lose this bacterial armor. And bullet point 3. If you give disease cells the bacterial signal, they immediately remember how to be healthy again.
Yes, perfectly summarized.
So, for you listening, ask yourself this. If the machinery inside disease cells is still perfectly functional, How close do you think we are to prescribing specific microbial signals instead of just suppressing the immune system? It gives a lot of hope, doesn't it?
It gives an immense amount of hope. Yeah. If we synthesize everything we've uncovered in this study today,
Bring it all together for us.
the gut is not just a tube. It's a highly specific regionalized terrain. In the upper regions, your microbiome produces nicotinic acid.
Right.
That nicotinic acid unlocks the PPAR alpha receptor hidden inside your human cells. This triggers the production of ketones, giving your cells the energy to build a thick, localized protective shield.
Immune barrier.
Right. And losing that bacterial signal leaves the tissue directly vulnerable to devastating injuries like Crohn's disease.
It fundamentally changes our definition of the human body.
It does.
We are not a self-contained organism. We are a symbiotic superorganism. Your day-to-day health, the literal physical structure of your organs, and your resilience against disease, they're all hyper-local phenomena.
Yeah, you rely entirely on microscopic bugs to hand your human DNA the instruction manual on how to behave.
It is a profoundly humbling reality. Our structural integrity is basically on loan from our microbial tenants.
Which leaves us with a provocative thought to chew on as we wrap up this deep dive.
I love a good provocative thought. Lay it on us.
I want you to consider this long after you finish listening today. If our gut bacteria are secretly managing the physical cellular architecture of our colon, region by region, literally turning our human genes on and off,
Yeah.
what other parts of our body's physical structure are being built, maintained, or neglected by microscopic tenets we barely even understand yet?
Oh, wow. Are they pulling the genetic strings in the skin?
The lungs.
The brain. It really makes you realize we are just scratching the surface of the biological city operating inside us.
We really are.
Well, thank you for joining us on this deep dive. The next time you think of your digestive tract, I hope you never picture a simple plumbing pipe ever again. Keep questioning, keep learning, and we'll see you next time.